This in vitro assay measures mycobacterial metabolic activity in whole blood to evaluate the effect of interactions between mycobacteria and host immune cells. First, add reporter gene-tagged mycobacteria to whole human blood in order to provide a real-time readout. Over a time course, collect sample supernatants for cytokine measurements and cell pellets for RNA analyses. Continue to lyse the red blood cells and, using serial dilutions and bioluminescence, measure metabolic activity of the mycobacteria. Finally, calculate a ratio between the final bioluminescence signal, expressed as relative light units, and the bioluminescence signal at the time of inoculation. The results indicate the host's ability to contain mycobacteria.
This whole blood reporter gene assay can help answer key questions in the field. For instance, does the presence of certain effector immune cells in a blood sample have a functional impact on the organisms, in this case mycobacteria?
The main advantage of this technique over existing methods is that the enumeration of mycobacteria can be obtained in real time, rather than counting colony-forming units after 3 weeks of incubation. An exact and reproducible inoculum can be determined each time the experiment is conducted.
First, grow a BCG lux culture to mid-logarithmic phase. Using the luminometer tubes, prepare duplicate samples of 1 ml of a 1 to 10 dilution of BCG lux culture in sterile PBS. At the luminometer, load the N-decyl aldehyde substrate tube and prime the